In this video we will discuss about how this stiffness affect the glycocalyx expression. So we have discussed a part of this in our previous video. We will continue our discussions from the point that where we have stopped last time. So in the last video we have discussed about two glycocalyx related proteins, that is heparin sulfate and hyaluronic acid. Here the researchers also shown the results about glycosylation. So in the previous video the quantifications was done on the fluorescence of the heparin sulfate and hyaluronic acid. However, in this case for the glycosylation one, which is one of the core protein for the glycocalyx, here the quantifications are done based on qPCR, and in the and they have checked the mRNA levels. And so what they have find out is that in these different endothelial cells, that is BAEC, they are able to see the glycosylation mRNA is high. Not high, it's like statistically non-significant. It looks like high. However, when they compared the statistical analysis, when they did the statistical analysis, they are able to see that the increase is not statistically significant for these BAEC endothelial cells. But for the MAEC endothelial cells, they are able to see the mRNA, glycosylation mRNA goes down significantly compared to the 2.5 kilopascals in the glass and also in the 10 kilopascals hydrogel. Polyacrylamide, they have used polyacrylamide based hydrogel coated with fibronectin. And for the band 3, the similar results they have seen that compared to the 2.5 kilopascals, in the case of glass, the mRNA level of glycosylation mRNA goes down significantly. So from that, what they conclude from these results, and what they conclude that in general, with increase in the matrix stiffness, there is a reduction in the glycocalyx expression, or there is a reduced glycocalyx has been observed in different endothelial cells, and that can lead to the altered cell phenotype. And this could be one of the key point that needs to be considered whenever someone is studying the effect of glycocalyx on any biological system in the in vitro model. So this stiffness considerations must be taken care of. So what they finally conclude is shown in the table that is presented on the right hand side. So they have not just tested these three cell lines, but they have also tested for HUVEC and RFPEC. And what they are able to find out that for the heparin sulfate, it is reduced in the glass for this BAEC, MAEC, band 3, and even reduced for this glass plus stiff gel for the HUVEC. But for the RFPEC, it is reduced only on the stiff gel. And these comparisons are done with respect to the soft hydrogel that has around 2.5 kilopascal. For the hyaluronic acid, they are able to see that only in the case of BAEC and the hyaluronic acids reduced, and for the case of stiff gel, the RFPEC reduced. But for the case of MAEC band 3 HUVEC, there is no change has been observed. And for the glycosylation one, what they see is that there is no change for the BAEC. This can be seen on the left hand side, and it is reduced for MAEC in the case of glass and stiff gel. That is shown, that can be seen on the left hand side. And for the band 3, it is reduced for the glass only. And for the HUVEC and RFPEC, for the HUVEC it is reduced for glass and stiff gel similar to the MAEC, and for the RFPEC it is reduced only for the stiff gel similar to the band 3. So all these results made them to conclude that majorly there is a reduction in the matrix stiffness when in the endothelial cells. So what they conclude is that majorly because of the matrix stiffness, there is a reduction in the glycocalyx formation or glycocalyx expressions in the endothelial cells, and that can lead to the altered cell phenotype. So this is what they finally conclude. I hope you understood this small paper, and it's easy to understand the results that they are presented in this figure or in the whole paper. If you need to understand more about their experiments in detail and wants to refer to the supplemental figures, you can go to the reference that has been provided in the description. So that's all for this video. Thank you for your time.